helped to change medicine from a field that was steeped in theories about humors and vapors to have its modern underpinnings, and helped create the modern field of immunology. Now, the second half of the 19th century is one of the most remarkable periods in the history of biology and medicine. So as most of you are aware, in 1854, John Snow discovered that cholera was actually transmitted through the water supply. In 1859, we have the publication of The Origin of Species. In 1865, an obscure monk, a Moravian monk, wrote a treatise on the hybridization of peas. And in 1873, Robert Koch, who was Emmy Fratz's husband, he was then the chief medical officer in Wulstein, which is a small rural town in Germany. And he created a laboratory in his little four-room house, using as a centerpiece the microscope that Emmy had bought him for his 28th birthday. And it was in this laboratory, in this small town in Germany, that he discovered the bacterial origins of anthrax. And as most of you know, he would go on in his life to discover the bacterial origins of tuberculosis and cholera, among other things. By the early 1880s, most of the exciting work that was coming out in the field of medicine and biology in general was coming out of two powerhouse laboratories. There was a laboratory of Koch in Berlin. He had moved by then to Berlin. And there was the laboratory of Louis Pasteur in Paris. And the pace of discovery was truly remarkable. To just cite one example, in 1884, Loeffler described the bacillus that causes diphtheria. In 1888, Roux and Yersin showed that supernatants of the culture of this bacteria contained a lethal toxin. In 1890, Brigger and Frankel, working with Koch, showed that injection of this lethal toxin into guinea pigs could create some form of immunity. And later that same year, in Koch's laboratory, Shibesaburo Kutasato and Emil Behring chemically inactivated the diphtheria toxin. So they took supernatants of these toxins, treated them with a chemical called iodoform to inactivate it, and they were able to inject increasing amounts of this modified toxin into large animals and produce something in the serum of these animals, which they called an antitoxin. Within a year of this discovery, there were children who were dying, who were gasping for breath with diphtheria, who were being miraculously saved by the injection of this antitoxin. Now, around this time, a young man called Paul Ehrlich, who had actually left Koch's laboratory for a while and had gone to Egypt, where he was recovering from tuberculosis, returned to the laboratory. And when he returned to the lab, he saw that there was this wonderful discovery that had been made about antitoxins. And he very quickly realized that his colleagues had missed the big picture. There was something much bigger than this antitoxins that had been discovered, but there was an underlying principle, and the underlying principle was that foreign substances, which he went on to call antigens, would induce the formation of some complementary molecules, which he called anticorpus or antibodies, and that this was an underlying principle of how the immune system worked. Ehrlich should be considered as one of the geniuses of biology, someone that we should think of in the same breath as Darwin. Darwin was right about one great thing. Ehrlich was right about half a dozen times. He thought about autoimmunity. He predicted the existence of autoimmunity. He would go on to come up with a theory of tumor immunology, which we still accept. He discovered the world's first chemotherapeutic. He realized that serum therapy wouldn't do everything, so he actually generated the world's first chemotherapeutic. So everyone owes that debt to him. But I think his greatest contribution was 1896, when he came up with a theory of the system. And you need to remember that this is about 65 years before we have conceived of the fluid mosaic model of the membrane. This is more than 7 decades before the first hormone receptor, so first polypeptide receptor, would be discovered. And Ehrlich came out with a theory where he described cells, the immune cells, that we would not know what they are for 64 more years. But he described these immune cells, and on the surface of these cells, there were these structures that he called antibodies. And then he described the activation of these receptors by antigens, which would then cause some signaling event to occur, which would then result in these cells secreting this specific antibody for days to come. Okay? And this conception of how the immune system worked has stayed true. And if you think about immunology for the next 50 years or so, in reality, there was a lot of work that was done on antibodies, but nothing was really understood about how the immune system worked. And it was only about 1957, when Ehrlich was rediscovered by two different people, that we actually had some movement in the field. So by the mid-1950s, people had started to think about how the immune system worked. Again, I want you to remember that nobody knew what lymphocytes did at this time. So when Burnett and Talmage came up independently with the theories of clonal selection, they still didn't know what lymphocytes did. They knew there were some immune cells, that immune cells made antibodies. They were still thinking in terms of antibodies. And basically, Talmage's idea, which Burnett kind of borrowed, was that you had a bunch of immune cells with different receptors. Antigen came by and selected one of them. That cell would then expand, so this is clonal expansion. And then the cell would go on to secrete one type of antibody. And so this was clonal selection as was conceived. And this was the clearest theory about how the immune system worked, predicted by Ehrlich. So what we're going to do in this first lecture today is we'll talk about the functions of antibodies. We'll talk a little bit about B cells, how they're activated, the role of helper T cells. So Andy mentioned T follicular helpers. We'll talk a little bit about them. We'll mention therapeutic targeting of B cells. And then I'll try to summarize everything in some different format. Okay? Now, so what you want to remember about antibodies is none of us would be alive today without antibodies. No vaccine that actually works today works without making high affinity somatically mutated antibodies. It is essential. You need to go through the immune response, make really high affinity antibodies to protect against the viruses. So vaccines work because we make antibodies. CTLs, which we'll talk about in the next lecture, come to clean up. If you aren't protected by your antibodies and then you do get infected, then CTLs come into play. Obviously, helper T cells are important in the making of high affinity antibodies, and we'll talk about that. Antibodies, as you know, are also the mediators of many diseases. So a number of diseases are antibody dependent. So whether you think of type 1, type 2, or type 3 immune responses, when you think of immunopathology, antibodies are involved in them. And today, as all of you know, and for instance, Abul talked about PD-1 blockade, and we'll come back to that, antibodies are very powerful therapeutics. So antibodies are important from these various aspects. We'll try to use this lecture to try to understand how the immune system actually works, and we'll talk a little bit about antibodies. So just to remind you again, when you think about diseases involve IgE, and then we have antibody-mediated diseases. We call them type 2 diseases, where antibodies directly cause problems, if they bind to tissue antigens or to cells. And then we have immune complex-mediated diseases like lupus or serum sickness, where it is an antigen-antibody complex or mixing complement that is involved in the disease process. Okay? When Ehrlich and Behring studied antibodies in the 1890s, they had no concept of what these molecules actually look like. And it was only again in the early 1960s that we start to get a view of what an antibody looks like. So we think of these Y-shaped molecules, but they were only figured out in the 1960s, that you have these two chains. They form these dimers of the two heavy chains with one light chain bound to them. And so the general picture that most of you must have about antibodies, since this is now in high school textbooks, as I've noticed, is that you have these heavy chains, and these are shown in two different colors, but it's the same heavy chain over here. And associated with the heavy chain, each heavy chain is a light chain. Again, it's the same light chain and the same heavy chains in a given cell. And here we are showing you IgG and IgM. IgM has a few more domains than IgG, and we'll talk about the isotypes in a minute. What you also want to remember is that the binding part of the antibody, the part that binds antigen, is in these domains called variable domains. So variable domains have a lot of differences from one antibody to the next. The constant domains are pretty much the same in all antibodies. So it is the two variable domains that come together, and we talk a little bit about this structurally, that create the antigen binding site. So this is a picture of, this is a, where you're looking at the heavy chain in red, the light chain in green, and in blue is the antigen that it's bound to, and it's the FAB portions over here which are involved in binding, and it's the FC portion which mediates the effector functions of antibodies. Okay? And stop me at any point if there's anything that's not clear. So the point that you all should probably know by now is that the diversity of the antibody comes from these three sequences in the heavy chain, which are called CDR1, CDR2, and CDR3, also known as hypervariable regions 1, 2, and 3. And these are the parts of the variable domain where the sequence varies considerably from one antibody to the next. Okay? The rest of the antibody is pretty constant. And this is, we're coming in terms of the structure, and I'll discuss this in a minute. You have these three loops that stick out from each immunoglobulin domain in the variable domains, and those loops that stick out form CDR1, 2, and 3. And I'll try to explain that a little more in the next few slides. So the basic structure, and we'll talk about structure for just a few minutes, and it helps us to think about the T cell receptor as well, is that the recognition domain as well of an antibody are all Ig domains, which by nature have a very fixed structure. This is not unique to the immune system. But in an Ig domain, you should think of an Ig domain as a ribbon that is opened up, which you fold together. So you have this ribbon of beta strands, which you fold together to form a barrel-like structure. And if you put a disulfide bridge to hold these two halves together, you have basically the structure of an Ig domain. But between those strands, as you'll see in the next slide, there have to be loops. So these are the strands, and you have these two halves brought together with a disulfide bridge holding them together. And these loops are the ones that stick out on the top, and it is these loops that are going to contact antigen. So you can think of an antibody as being made of heavy chain and light chain, with three fingers sticking out at the top of the variable domains. And the three fingers are these three loops. And when you bring these 6 fingers together, they're going to form the structure that will recognize antigen. Okay? Now, if you are looking at a small antigen, for instance, like a small molecule the size of, let's say, a sugar molecule or a drug, the fingers come close together and form a little cleft, and they bind the antigen. But when you want to bind a protein, and this is how we should think about the T cell receptor, then those fingers have to splay out to form. So the T cell receptor, the surface will accommodate MHC and peptide. In this case, you have an antibody which is binding to a protein, and it is the splayed out fingers that form a big surface upon which a protein can bind. And this is shown in the next slide, where what you're seeing here is the FAB portion, the binding portion of an antibody, recognizing Hennig lysozyme. And all the residues here in red are those that are going to bind to these residues on Hennig lysozyme. And this glutamine over here is going to stick into this little pocket in this groove. So this is heavy chain, light chain, forming a surface which is going to bind to the antigen and hold on to it. Now, what you also probably know is that antibodies come in different flavors. You have different isotypes. So when you start an immune response, the B cell that is going to start the immune response has IgM and IgD on the surface. And then in order to have different effector functions during an immune response, T cells are going to instruct the B cell to go and switch to a different isotype so that the binding part of the antibody won't change, but then the effector parts are going to change depending on the type of pathogen that you see. And we'll discuss that in a little more detail. So the isotypes of antibody, IgG is just a tetramer like this of heavy and light chains. IgD, which is found on naive B cells, has also got a tetrameric structure like this. IgE is similar, and this is very important in allergic responses or responses to certain types of parasites like worms. IgM is a multimer. It has the basic tetrameric unit, but it has 5 of these brought together, so it has 10 binding sites, and it is joined together by an extra chain called a J chain. So an IgM molecule with these 10 heads, this is the antibody that we're going to make. Usually each binding site is of very low affinity in an IgM molecule, but because there are 10 binding sites, the avidity of the molecule for antigen is great. So you can just imagine that if you have something with 10 tentacles sticking out, if only one of them were to bind well enough and release itself, maybe another of the 9 will grab on. So effectively, by having multiple binding sites, even though the affinity of each binding site is low, you get a reasonably good binding and reasonably efficient clearance of pathogens. So this is the value of an IgM. Even though it's low affinity, you start off with it, it can still give you a protection because although it hasn't become refined in terms of somatic mutation and becoming high affinity, as we'll talk about, it does have multiple binding sites. Similarly, in the gut, we have IgA, which has 4 binding sites. There are 2 tetramers joined together by a J chain. And IgA, I'll remind you, is heavily glycosylated and is not easily degraded by bacterial proteases. So it's highly specialized to be at mucosal surfaces to give us protection from mucosal pathogens. Okay? So there are a number of functions of antibodies which I'll touch on very briefly. In terms of one of the effective functions is neutralization. And we don't use this in the Schwarzeneggerian sense of the term. Neutralization doesn't mean you're going to blow something up. It means you're just going to block some structure on the surface of a virus or on a toxin and block it from being able to bind to your receptors. So a virus cannot enter your cells or a toxin cannot enter your cells because an antibody binds to it and prevents entry. Okay? We call that neutralization. Very important function of antibodies. The term opsonization is where an antibody functions something like a specialized, kind of a butter. So you have a pathogen that's yucky and you don't want to eat it, but you coat it with an antibody butter. And now it looks really good to eat and the phagocyte eats it up. Okay? So basically, the Fc portion of the antibody sticks out from the microbe. Even if the phagocyte lacks a receptor to see the microbe, it can now recognize the Fc portions of the antibody sticking out, grab onto them using Fc receptors, internalize it, and this is called opsonization. Okay. Another function of the antibody is ADCC, and we'll come back to this in the next lecture. But basically, an ADCC is an NK cell mediated killing of an infected cell. If I have a virally infected cell and it's coated by an antibody, then a different kind of Fc receptor on the NK cell recognizes the virally infected cell and then bumps it off using perforin and granzyme. And that's called antibody-dependent cellular cytotoxicity. And finally, there's the function of complement linked to antibody. As you've heard from Andy, complement can work in different ways. One of the pathways involves antibody using complement as an effector mechanism. And what complement can do is at least 3 different things. You have lysis of microbes. You have phagocytosis of microbes opsonized with complement. To this, you should add, you also have the clearance of apoptotic cells. So complement is important in clearing apoptotic cells, but that's usually not antibody dependent, but it's complement dependent. And then finally, they have these fragments of complement which drive inflammation. So you have, every time you get the generation of C3a or C5a, C5a in particular can drive inflammation by functioning in part as a chemokine. So a quick overview of complement would be, and you heard this again from Andy, C1q binds, you have C1R&S, C1R activates C1s, C1s cleaves C4 and C2, and then you get the formation of a protease which can cleave C3. So you need to create a C3 convertase. And when you cleave C3, you get C3b, which attaches itself to microbes, and C3a, which is released. So the first step is you form a C3 convertase. The second step is you form a C5 convertase. Again, you get C5b, and then you have the release of C5a, which can drive inflammation. And C5b can help nucleate with C6 to C9 the formation of the C9 pore, which will form a pore in the microbial membrane, and then salt and water will go in and blow the bug up. Okay, so that's one of the basic functions of complement. Okay. If you have any questions about complement, I'm happy to go into more detail, but if it's something that you want to avoid, I understand why. Okay. Okay. The other thing, so the Fc portion of the antibody, one of the things it's doing is it fixes complement. Okay? And whatever it recognizes, if it's aggregated, it can fix complement. The other thing the Fc receptors recognize, as we briefly mentioned, are Fc receptors. And most of the antibodies which do effector functions are IgGs. So the Fc receptors for IgGs are called Fc gamma receptors, and they're numbered 1, 2, 3, and so on. Okay? Fc gamma R1, 2, and 3a, 2a and 3a, are all activating receptors. So they activate phagocytes. The phagocyte becomes better at phagocytosis, but it also can get activated to release things. It can release cytokines. Okay, so activation is occurring through most Fc receptors. The ITAM motif, does anybody know what an ITAM is? Have you heard the term before? Yeah. So an ITAM stands for immunoreceptor tyrosine-based activation motif. It's one of the most horrible names in immunology. But basically, when you see the A, it's going to activate. It has tyrosine residues that can recruit a kinase. Whereas the ITIM motif, which is an inhibitory motif, also has tyrosine residues, different sequences around it. And this is going to recruit generally phosphatases. And it's going to inhibit signaling. So Fc gamma R2b is found on B cells as well as on dendritic cells and macrophages. And it's basically an inhibitory receptor. So you need to dampen inflammation. You need to dampen B cell activation. And a lot of that occurs by antigen coated with antibody, which then recruits the activation or causes the activation of an inhibitory receptor, which dampens signaling. Okay? Now, the other few functions of antibody binding to Fc receptors, so there's a different kind of Fc receptor on epithelial cells. And epithelial cells want to take an antibody from the abluminal side of the gut, for instance, and transfer the antibody into the lumen where it can protect you. So you might make an antibody underneath your gut mucosa and you want to put it into the lumen of the gut to provide protection. And the receptor that does that is called the poly-Ig receptor. And basically what you have here is a plasma cell, let's say making an IgA antibody. You remember that the IgA antibody has a J chain. The epithelial cell has a receptor, which is, it's called the poly-Ig receptor, which can recognize the J chain, so it binds to the antibody. Then it endocytoses the antibody, the J chain coupled to the IgA molecule here, but it doesn't digest it in lysosomes. It takes this molecule that it captures across the cell from the basal side to the luminal side and then releases the IgA. So this process is called transcytosis, where an endosome does not fuse with lysosomes, but is trafficked to the apical surface of the cell where it can release its contents. Okay? And this is one property of the poly-Ig receptor. Now, Abul insisted that I will tell you this, so I have to tell you this, and that is about another Fc receptor called FcRn. Now, the real reason you should know about FcRn is not what I have on the slide. Okay, so FcRn, and I see somebody in this room who actually works with Alan Walker. So FcRn is really involved in transport of IgG across the placenta and across the infant intestine. Okay, so it's a receptor. It looks like a class 1 molecule, but it's a receptor that can take IgG across the placenta. And we know that IgGs can cross from mother to the fetus across the placenta. That's one of its very important functions. However, there's another function. It's found on endothelial cells. And IgG molecules live long. We make an IgG response. The IgG is there for a few months. And the reason why it hangs around for a few months and is not cleared like other plasma proteins in the liver is that it can bind to this endothelial receptor, which is FcRn. FcRn will take it into an endosome, allow it to go through some rest and recreation. So it's hidden away from the blood. So all our IgG molecules are constantly going into cells and hanging out for a while, protected from the ravages of what's going to happen in the blood. Then they get released again into the blood. So they just extends their lifespan. So most of the IgG molecules you make hide out for some of their life, then keep coming out, and this keeps them long-lived. So if you put an Fc portion onto some other protein, you can give it a long life. So that's the reason why some therapeutics have the Fc portion of IgG added onto them just to make them longer lived. Okay? And the last function of antibodies that I should mention is IgE. And I think most of you know that IgE does not float around in the blood. The moment it gets out of a plasma cell, there are receptors for IgE, high affinity receptors on mast cells and basophils. And the mast cells will just grab on to the Fc portion of the IgE, and they will be coated by the IgE. So the mast cell takes up whatever IgE you make, that you don't find so much in the blood, and has it on the surface, and it waits for a worm or an allergen to come by, which will then cause cross-linking of these receptors, degranulation of the mast cell, then you sneeze, or you get anaphylaxis, or you get something terrible, or you get asthma, you get responses because of IgE being triggering the IgE receptor which is bound to it, causing degranulation. Okay? Do you have questions so far? We've covered some very fundamental things about the structure and function of antibodies. Any questions? It's clear? Okay, then I'm going to summarize this part of what I've said. Antibodies protect you, but antibodies kill. Autoantibodies can make you very ill. Antibodies coat pathogens. They coat the infected cell. With NK cells and phagocytes, we give those microbes hell. When antibodies fix complement, the shit really hits the fan. You'll be blown to bits, little microbe. Run away if you can. You take a set of beta strands, you get a beta pleated sheet. Fold the pleated sheet in two, and the barrel is complete. Clip it with a disulfide bond, you have an Ig fold. Found even in archaebacteria, this domain is old. Loops make connections between the beta strands. If you want to make an FAB, please use both your hands. The loops are like fingers. They stick out at the top. Complementarity determining regions. The cream of the crop. 3 fingers from the heavy, 3 fingers from the light come together to create an antigen binding site. Bring the fingers close together, you get a cleft with a purpose. Splay the fingers widely, you get a protein binding surface. Antibodies protect you, but baby, antibodies kill. Autoantibodies, they make you very ill. Oh, you Y-shaped globular proteins with the name that Ehrlich gave. If the good doctor could hear this doggerel, he'd be turning in his grave. Okay. All right, so, oh, thank you. Applause is only for a bull, okay? Not for me. So now, during an immune response, we're now going to go a little bit into how you make antibodies and what happens in an immune response. And I just want to remind you that we have a thing called a primary immune response. You first expose somebody to antigen, the naive B cells get activated, they differentiate into plasma cells, The plasma cells secrete some antibodies, initially some IgM, maybe a little bit of IgG. Then you immunize again, as we typically do in immunization schedules. You get more memory cells now, and these memory cells then differentiate into plasma cells. You get higher levels of antibody production. At this point, your immune response is somewhat refined. The antibodies are going to be higher affinity. More of them have switched to IgGs or maybe IgAs or IgEs. So this is called a secondary immune response. Okay? The primary immune response, not so much memory. The secondary immune response, a lot of memory. And at the end of the day, you're going to have plasma cells, which are long-lived and are going to be factories secreting antibodies for your lifespan. And then you have memory cells, which are hanging around, waiting to wreak vengeance if the pathogen seeks to come back. Okay? And that's the big view of the immune response. Now, in order for all of this to happen, you need antigen to be delivered to a B cell. And as you've gathered from Abul's lecture, all the activation of lymphocytes is going to occur in secondary lymphoid organs, in lymph nodes, in the spleen if it's a blood-borne pathogen, in Peyer's patches if this is the gut. So how does antigen get there? So there are a number of ways in which antigen is going to reach the follicle. Remember that the B cell zone in the lymphoid organs are follicles, and then you have separate T cell zones. So there is a sinus. So afferent lymphatics arrive at the subcapsular sinus, which is basically a little cavity around the lymph node. And in the subcapsular sinus, they can be taken up by these macrophages, which are called subcapsular sinus macrophages. So there are specialized macrophages that can capture antigen and take it to a follicle. If the molecule is small, if it's, say, a protein of less than about 70 kilodaltons, there are little conduits from the subcapsular sinus which go straight to the lymphoid follicle. So a small protein antigen, if it arrived in debris from the tissue, so you had an infection somewhere, from there you had all this crud coming through the lymphatics. If it's a small protein, it will make its way through the conduit and it will reach the B cells over there. Some of the antigens which are neither captured by these two mechanisms will go through into the medulla of the lymph node, and their specialized dendritic cells, conventional DCs, may capture the antigen, not chew it up as they would do for T cell activation, take it on the surface and go to a follicle and allow the activation of B cells. So there are a number of ways in which an antigen may reach its cognate B cell. Yeah, so there are many different types of dendritic cells, and I will go through a nomenclature issue now that you bring it up. What you need to remember is there are conventional dendritic cells. Those are the dendritic cells you've been hearing mainly about. They activate T cells. Some subsets of them can activate B cells, and that's what we generally call dendritic cells. Someone says dendritic cells, that's what they mean. You should call them conventional dendritic cells, or if you want myeloid dendritic cells, but conventional dendritic cells is the better term. There is another cell type, which is extremely important in many diseases, called a plasmacytoid dendritic cell, which its origins are somewhat controversial, but I would say it's a completely different cell from a conventional dendritic cell. Its specialized goal is really to protect you from viral infections. It makes a lot of type 1 interferon, and it can convert itself into a cell that does some of the functions of conventional dendritic cells. So that's a PDC. And then there's a third cell, which you should, in the same breath, say, I know the name of the cell, but you should be able to say, it's not a DC. Okay, it's called a follicular dendritic cell. It is nothing like a dendritic cell. It has the name dendritic to it. Convention forces us to keep calling it a follicular dendritic cell. It is not from a blood origin. It is not from the bone marrow. And it has a completely different function, which we will come to a little later. So we call that an FDC, and let the D be always only a D and never dendritic. Okay? So it's not a dendritic cell. It's an FDC. So three types of cells, you should know that there are three different cells. Very different cells. Now, among dendritic cells, there are subsets. There are dendritic cells which are in the tissue, and dendritic cells which go into lymph nodes, dendritic cells which don't migrate. So there's some differences in dendritic cells, but we still will call those conventional dendritic cells. Okay? Maybe that's a little too much, but if you can remember these three different things, and they're all different, and they have very different functions, that's key. Yes?
So my understanding was that B cells were closer to the T cell cells when they got activated and went up to the follicle after they got activated. So am I misunderstanding?
Yes, it's a misunderstanding. It will be clarified shortly, at which point you can come back to me if it's not clear. I hope I'll make it very clear. Yes?
How close antigens get into the lymph system, and are they just little peptides that are floating along, or are they?
Okay, so you got an infection in your little finger. Microbes got under your skin and started to multiply. Okay? Let's say they died there. Lots of them will die. Lots of them will be eaten up by neutrophils, but there's going to be debris from them. So the microbe is now chewed up into bits. Okay? The innate system does some good. It doesn't get everything, but it gets some things chewed up. Those bits and pieces have to go somewhere. They're proteins often, or lumps of membrane. They can't go back across the endothelium. Okay? They are in the tissue. And the tissue, we have a separate drainage system. It's called lymph. Okay? It's stuff outside blood vessels. It's going to enter these open-ended lymphatics. And the lymphatics from my little finger are going to travel a long way, all the way to my armpit, to the lymph nodes over there. And then I'm going to get this swelling in my armpit because of this cut in my little finger. Those antigens that were left out, or some were captured by DCs, some were not captured by DCs, they go draining in all the way to the lymph node. And that's where you're seeing them coming in through the afferent lymphatic. Okay? Contains everything: proteins, pieces of membrane, entire bacteria, fragments within DCs, they're all coming into the lymphatic. It's a clearance system. Okay? Any other questions? Okay. So now imagine that I am a one-armed bandit, okay? And I have only one arm. Okay? So I want you to think of that as an analogy. And the second picture you need to have of me is that I'm actually a deity, and I have 10 arms. Okay? And these are two very different things. So if you want to understand how you activate a B cell, you need to think about this analogy, and we'll come back to it. So if I have a B cell that is going to be able to see a monosaccharide or digitonin or digoxin or some small molecule, okay? It has a binding site, it has high affinity. The antigen receptor is an antibody, it can recognize this thing really well. So whatever this little thing is, it's recognized by an antibody. We call these little molecules antigens. There's no question it's an antigen. But I've already told you there's no signal, right? It can bind to the receptor, but it cannot cause the receptors to cluster together, and therefore it can't activate them. If I took this little monosaccharide here, and if in nature or artificially I polymerized it to make it multivalent, okay? So now I have the same unit coming 25 times or 100 times. In that case, this structure can cause my receptors to come together, and when the receptors come together, they're going to signal. So the analogy I wanted to use for you is if I'm a one-armed bandit, I can just grab one thing at a time. I can grab Chopin, but then I have to drop him if I want to grab Michael. Okay, I can only do one thing at a time with one arm. But if I have 10 arms available, then I can grab 10 of you, grab all of you together at the same time and bring you all together, and then maybe you'll make music. Okay, and that's how signaling works. If you are multivalent, you can cluster receptors because they're all linked together and then cause them to cause signaling to occur. If you're monovalent, you can bind, but you can't activate anything. So this brings us to some basic definitions. So haptens are small molecules. They are antigens, but they are not immunogens. So the small molecule can be recognized, but it can't cause immune activation. Okay? So we call those molecules haptens. And the laws here are all immunogens are antigens, but all antigens are not immunogens. Okay? And that's a must. You have to know that. Memorize it 10 times tonight before you go to bed. Okay? Then this course will be worth the money. If you can remember that one thing. Okay, so the B cell receptor, you start off with the unit. It is made up of heavy chain and light chain attached to the membrane. It has two associated chains with the horrible name of Ig alpha and Ig beta. These are not antibodies. They are just associated chains, and they contain ITAMs. They have these tyrosine-based motifs which are going to allow signaling to occur. What's shown in blue here is a kinase, a SAC family kinase, part of the B cell receptor for signaling. When you add an antigen that can cross-link these receptors, now you see that those two kinase molecules have been brought close to each other. They can phosphorylate each other, and they can introduce these bulky phosphate residues that will decorate the tyrosines. So you now have this structure showing up with these bulky tyrosines. You can imagine that somewhere on the ceiling there was nothing over there, and suddenly a big bulge appeared. And I'm a protein inside the cell. I see that bulge for which I have a cleft that can bind to it, and I can go grab onto it. Okay? And that's what's going to happen in signaling. SYK is waiting with these two clefts in it, but it waits for these bulky phosphates to appear on the ITAM, and when that appears, SYK can be recruited to the B cell receptor complex, get activated. SYK itself is a tyrosine kinase, phosphorylate downstream targets, and induce B cell signaling. Okay, and basically that's what happens in signaling. You create some novel shape on the inside of the membrane. A protein that can be recruited to the new shape now comes into play, grabs onto the inside of the membrane, and signals. Okay? So in order for signaling to occur, now you have the concept of, does everybody know the difference between a co-stimulator and a co-receptor? This is just broad immunology. What's a co-receptor and what's a co-stimulator? That is not a definition. So I was told that CD4 is a co-receptor. That is correct, but that is not a definition. That is a description of a fact. I wanted to know what is the definition of a co-receptor, even though you're right, and how do you discriminate between a co-receptor and a co-stimulator? This is just a conceptual issue. Not actually correct. So I hear that co-receptor doesn't signal. No, it actually helps signaling, and it's got things attached to it that will do signaling. So what's the definition? You heard about co-stimulators this morning, so I just want to make sure you get the concept clear. Co-receptor is CD4, you're right. Co-stimulator, what is it? What's the difference? Say that again. The ligand for the receptor is antigen here. We're talking about antigen receptors, right? So the ligand is antigen. There's a ligand for the co-stimulator also, but that's different. So maybe you heard signal 1, signal 2, and all that stuff today. So I just want to make sure you clarify this. Yes? Abul's not here. I can go on for another hour. That's okay. You're right. The terms are correct. So the co-receptor helps signal 1. It helps the antigen receptor deliver its signal. And the co-stimulator is nowhere near the antigen receptor. It doesn't even have to be in the same complex. The co-receptor must be part of the antigen receptor complex that recognizes antigen. It must be physically part of it, and it must help the generation of signal 1 through the antigen receptor. The co-stimulator is some other receptor on the cell which functionally helps the lymphocyte to be activated. But it doesn't have anything to do physically or functionally directly with the antigen receptor. Okay, so that's the distinction you want to make. So the co-receptor for T cells that you've heard about all morning, one of them is CD4. CD4 helps the T cell receptor to signal. It actually brings LCK and helps it to signal. CD8 is a co-receptor for the other cells. We'll talk about it in 10 minutes. co-stimulators, you remember from the morning? CD28, big co-stimulator for T cells. So in the case of the B lineage, co-receptors that we will talk about is complement receptor type 2, we can define as a co-receptor. Okay? It is actually something that if the antigen has complement bound to it, like C3d, the co-receptor here is CD21 or complement receptor type 2. It comes into play because it's bound to the same complex of antigen over here. It brings with it kinases which can activate the B cell receptor and induce signaling. Very similar to what CD4 does. There is a broad equivalent of what a co-stimulator would do would be the toll-like receptor here on the B cell, which can provide signals. It's nothing to do with the antigen receptor complex. Okay? You can think of it as some sort of a co-stimulatory signal, whether we define it as one or not, but it's not a co-receptor. It is going to provide additional signals which may help the B cell get activated. So this is how you would activate a B cell. It's a sensing danger both from the complement, which is actually part of the co-receptor complex here, and also by TLRs, which are sensing TLR ligands from the microbe and so on. Okay? So to go through this quickly, we won't spend time on this, but I just want to tell you there are different types of B cells. Okay? We don't have to spend time on it today. There are follicular B cells, there are marginal zone B cells, and there are B1 cells. And these guys mediate mainly T-independent immune responses, coming to you in a sec. And the follicular B cells mediate both T-independent as well as T-dependent responses. And your question?
Like, what about CD40 and CD20 ligands? As per the classification for
Yeah, if you want to classify it, then you would call it as a co-stimulatory molecule. But I wouldn't normally put it right there. Okay? It is a way in which, through T cell help, you're going to provide another signal to a B cell during T-dependent activation, which we are going to come to the last few minutes of this talk. All right, so in a T-dependent, so from now on, the rest of this lecture for the next 5 or 6 minutes is about T-dependent immune stimulation. In this case, we have a T cell, an antigen is presented by a DC to a helper T cell in the T cell zone. Okay? You get this T cell to get activated and expand a bit, and then the T cell, when it's activated, is going to express less CCR7, more CXCR5, and start to migrate towards the B cell zone or the follicle. Okay? At the same time, there's some rare B cell that could recognize an antigen from the same pathogen, and some epitope on that antigen activated some specific B cell. This mild activation of the B cell, no proliferation is going to occur now, allows this B cell to express a little more CCR7, to ingest the protein, and to migrate towards the T cell zone. Okay, so you have an activated T cell going towards the B cell zone, and a slightly activated B cell going towards the T cell zone. And they meet somewhere in between. And what's really going to happen is that in this case, the B cell receptor is functioning as an endocytic receptor. It's going to grab the protein antigen, chew it up into bits, put a specific peptide from within the protein onto a class 2 molecule, take it to the cell surface. If this class 2 peptide combination is recognized by this T cell, this activated T cell is going to interact with it. The activated T cell expresses CD40 ligand, which was induced during activation, and then whacks the B cell through CD40. And it is the CD40 stimulus that is going to cause this B cell now to proliferate and expand. So this is a T-dependent response. It is not dependent on cross-linking the B cell receptor with a multivalent polysaccharide or a nucleic acid. It is dependent on a protein antigen providing a peptide to allow a T cell to recognize this B cell, and then activation is going to occur through CD40 to drive this response. So if I have to go through this again in terms of steps that are going to happen, you have activation of a helper T cell by a DC, expansion of the effector T cell, that is the T cell, CD4 T cell that's been expanded. It moves towards the B cell zone, a B cell moves towards the T cell zone, you have the first TB interaction, which is going to cause the T cell to cause this B cell to proliferate and differentiate. As a result, the B cells are going to expand. And some of these B cells, a few of them will even become plasma cells, and they won't be long-lived. They'll be short-lived plasma cells. Some of these activated B cells decide that they want to reciprocate the love they have seen from T cells and provide a signal back to the T cells. And so now we have this previously activated T cell being activated now by a B cell. And this B cell mediated activation of the T cell is crucial for this T cell to differentiate into what is going to be called a T follicular helper cell. So a T follicular helper cell is a polarized CD4 T cell that first was triggered by DCs, then got triggered by activated B cells using ICOS ligand, and that caused them to differentiate into these cells which have huge amounts of CXCR5, which can then, coming back to the question I was asked before, allow these specialized T cells to migrate into the follicle. So the T cell is now burrowing into the B cell zone. Okay? This is happening late. It's only after this B cell induced T cell activation. This activated T helper cell, this T follicular helper cell, is going to orchestrate the formation in the follicle of a novel Darwinian structure called the germinal center. Okay? So you have the formation of the structure where you have B cells. So you have one of those B cells was also called in, becoming almost like a tumor, proliferating more rapidly than any cell in the body, dividing every 4 to 6 hours. All this is orchestrated by the T follicular helper cell. So the B cell proliferates in what is called the dark zone. And as it proliferates, each of those progeny cells are going to go through a process of somatic mutation where the heavy chain and light chain variable regions are going to be mutated, heavily mutated. It's something the body hates to do, but it does it aggressively in the dark zone. We are fighting mutation everywhere because it's dangerous, but in the dark zone, we create mutation. We diversify the receptors. So you have genetic diversification, natural selection, and survival of the fittest. So the highest affinity B cells that come out of the dark zone get selected by T follicular helper cells, but they receive antigen from an FDC. Remember, I'm not using the word dendritic now. Okay? The FDC holds up the antigen. The B cell, which is of highest affinity, can take the antigen, present a peptide to a T follicular helper cell, and it will live. All its unfortunate sisters will die. Okay, so you have massive apoptosis of lower affinity cells, and only the highest affinity cells are allowed to live, become memory cells. They would have gone through class switching as well in the germinal center. So they've changed their isotype, and they now express a high affinity B cell receptor because it's gone through somatic mutation, and the whole process is called affinity maturation, where the highest affinity cell gets selected. And then it's going to emerge as either a memory B cell or a plasmablast that will migrate to the bone marrow to become a long-lived plasma cell. Okay? And that is the crux of the T-dependent B cell response. Have I answered your question about what happens to B cells that you'd raised before? So the B cells, one of those B cells is going to go back into the follicle with the T follicular helper and form this cancerous growth called the germinal center, where one cell proliferates like crazy, gets mutated, and then gets selected based on affinity. If you have any doubts about this, please ask me. Yes?
Not doubting you, but I'm asking, the T follicular helper cell, is it coming from what was a Th1 or Th2, or?
No. Th1, so there's a decision made to either become a Th1, Th2, or Th17 if the affinity is low. If the affinity is high, first choice, remember that you're going to live only if you make those antibodies, however much you might get excited by Th17s. You have to make a B cell that is going to go through somatic mutation. So the high affinity cells induce a specific transcription factor called BCL6. The low affinity guys don't, they make blimp. So the low affinity guys go into the other pathways. The high affinity guys all become T follicular helpers. If you didn't make T follicular helpers, you won't make the high affinity response, you won't be protected from the virus the next time.
I'm sorry, so you lost me for a second. So the high affinity T cell?
The high affinity CD4 T cells, the best affinity guys become T follicular helpers. The low affinity guys now have the option to become TH1, TH2, or TH17. Now, these high affinity guys, because the pathogen makes some, let's say it makes some IL-12 in the vicinity of the DC that activated the cell, that cell might go and become a T follicular helper, which makes a cytokine that drives switching to a particular, much like a TH1. So the cells that drive switching are not TH1, TH2, and TH17. They are T follicular helpers, which may borrow some properties of TH1, TH2, or TH17. Okay? If that makes sense. Right? Any other questions? Yes.
And the affinity variable, how is it defined?
The affinity means I couldn't fit, I'll show you a slide, I couldn't fit well and now I fit so well, I will never let you go. That's high affinity. It's absolutely conformational, created by the heavy chain and light chain both. It's selection of the highest, best conformational fit. So you've mutated both and you're just testing them out. I mean, from one B cell you probably make a, many, many thousands of which only one is really high affinity and you pull that out. Yes? Oh yes, so class switching actually starts even before you go into the germinal center. Okay, so class switching can start earlier, but the T follicular helper cell is going to help everything, IgM, IgA, IgE, any switch, it's going to help in the germinal center. Okay. Right, so I have a couple more slides. So just to remind you, switching, different isotypes are dependent on different cytokines, still T follicular helper making them. Interferon gamma will give you more IgGs, IL-4 will give you more IgEs, and so on. And in switching, what you're doing is you have this unit here. This is the variable domain, the VDJ unit here, upstream of mu and delta. You want it to, so this would make IgM through mu. But you want to put this VDJ, let's say you want to put it next to IgE. And so in the switching process, what you're going to do, you're going to break the DNA here, you're going to break the DNA there, and put this VDJ next to epsilon. So this is basically the goal, is to get the switch to go from this part of the DNA should now be brought close to this DNA. And how that happens is by a looping out method. So basically the chromosome is going to loop out. The VDJ unit is going to be brought from close to the mu as close to the epsilon. You have a double strand break here, a double strand break there, a loop, joining these two pieces together. So now the VDJ unit, which was next to mu, has now been placed next to epsilon to make the same binding site, but have a different effector domain. Okay? And that's only for the heavy chain. And affinity maturation, basically what you started out with is initially you had a low affinity antibody, like this round whatever in a square whatever, over here, not really fitting. But you went through somatic mutation, you put in new bases into the variable domains of heavy and light chain. Now you've got a really high affinity antibody that binds more tightly. Okay? And so that's the process that's going to happen in the germinal center. Essential, okay, in order to get a good immune response. You all know that you can deplete B cells by plasmapheresis. A very important method now clinically is using Rituxan. We deplete B cells using anti-CD20. Works great in some diseases which are caused by T cells. Okay? Multiple sclerosis is one example. There are a bunch of diseases now. Type 1 diabetes is a little less than multiple sclerosis, but where you deplete B cells and the T cell mediated disease goes away. And that is because you do not make CD4 memory without B cells. The best APC when you start an immune response is a DC, but the DC dies in 2 or 3 days. The cell that is going to present antigen subsequently for you to get memory is a B cell. Okay, so B cells are essential for memory, and you can take away B cells and many diseases will improve. IVIG is also a therapeutic. It basically dampens inflammation. BAFF antagonists have not been that successful. BAFF is a growth factor for B cells and follicles. So far, not great. They exist. They've been tried in lupus, and they don't work that well. So IVIG basically induces the inhibitory FC receptor, which is going to dampen inflammation. Okay? So finally, to give you an overview of B cell activation in a T-dependent response, we have activation of the B cell by a protein antigen. Helper T cells help in the process, without which this won't happen. You get clonal expansion. At the end of the day, you can get long-lived plasma cells, class-switched cells of memory isotypes, and they're all higher affinity. And the memory cells are the ones that will drive a secondary immune response. So when you vaccinate, you want to make sure you get an IgG response, because you want a memory IgG cell, so that the next time you see the pathogen, you'll have enough of a memory response to generate antibodies. Okay? And I'll take questions before I go to something else. I know I'm late, but it's okay. Yeah. Questions? Yes?
How does it decide which classes to switch to?
So if I have an immune response, so when you think of your Th1, 2, and 17, you think that the certain milieu drives an immune response to, let's say, get macrophages activated so you can swallow a mycobacterium, right? So you're going to make IL-12 to make a Th1 response to swallow the mycobacterium. So that same milieu can affect the T follicular helper and tell it, please go ahead and make a switch based on interferon gamma, which is going to allow me to switch to an IgG. The T follicular helper is going to make the cytokine that resembles that made by, let's say, Th1, Th2, or Th17, though it's not the only cytokine. There are different combinations. Th1, Th2, 17 is a bit too narrow. There's broader choices. And the cytokines that are made by the T follicular helper are going to say, you know, this is an immune response against a microbe in the gut, make an IgA. Or this is an immune response against a worm, make an IgE. And that's how it's going to happen. Yes?
Not all B cells are created equal. So my question for you, two-part. B-regs, the role of B-regs. And the second is, after B-cell depleting therapies such as anti-CD20, you get repopulation of different B cells at different rates. Can you elaborate on that?
Yeah, so one of the reasons why, so I'll come back to B-regs in a second. One of the reasons why CD20 therapy actually works relatively well when it does work is that B cells come back slowly. And when you come back eventually, the cells that are going to come back first are transitional cells. Okay, so transitional cells are the earliest B cells that can come out. But you often see in patients, we study a bunch of patients at MGH, where you often see them quickly becoming, you know, the type of rogue B cell like a plasmablast in certain disease and so on, which is caused by the milieu in which the antigen is still around, the disease is still around. So you might see different changes. Now, B-regs, which are very functionally defined, there are some good examples where B-regs seem to do something important. But nobody is yet able to give you a tight definition of a B-reg in terms of surface markers, which does not overlap with many other cell types. So B-regs overlap with transitional cells. They overlap with cells that we've called T2MZP cells. There's a lot of overlap. So no one has been able to do the clean experiment of depleting just B-regs. And so far, in the mouse field, no one has conditionally taken away B-regs in the cleanest possible way. So I would leave B-regs as an advanced course question where we still don't fully understand how and when they work, and whether therapies like B-cell depletion are actually getting rid of the B-regs as well, which they might do sometimes. So there's some uncertainty yet as to, though there's a lot of literature saying that they are important, and clearly they are important in certain disease models, more so than in humans. Though there's a good paper in lupus, at least, that addresses a possible role for B-regs. I don't think we've answered the question yet in terms of understanding what exactly they do and how they manipulate. They're supposed to make IL-10. They're IL-10 producers, and they're supposed, but IL-10 is also made by Tregs. Yeah, so the question is, when is the Breg so important that it is more important than a Treg? And that's not very clear. Okay. Any other questions? I can do the wrap later. Okay, we can. Yes.
Do lupus patients have more something to do with their memory B cells which they have accumulated over a period of time or something?
Say that again.
The lupus patients,
Yes.
do they have any information on how these memory B cells may have accumulated against certain pathogens in lupus patients?
No, okay, so I guess we do understand something about lupus. In lupus, you can assume that the B cell response is activated by nucleic acids, and that the B cell response is driven by the failure to perhaps clear apoptotic bodies, and the induction of that B cell response with a lot of TLR stimulation allows the generation of long-lived plasma cells which are directed against some chromatin component. Okay? And because they're long-lived plasma cells, therapies like Rituxan, CD20 depletion often don't work well in lupus. Okay? Because those long-lived cells don't have CD20 on them. You can deplete your naive B cells, nothing's going to happen. I think that's all we understand about lupus. There's lots of hits from GWAS. The tolerance effects, whether really there's a tolerance defect that can be proven to be correct in lupus is uncertain. There are many attempts to say that there are genes that are affecting tolerance. I would say so far we really don't understand it. And it's still a long way off. This is something many of us are interested in, but we don't have an answer. Yes? Yeah, so a T-dependent, so even all autoimmune diseases in which antibodies are important involve class switch mutated antibodies. Without exception. The antibody that causes the disease is usually an IgG and it's high affinity. So yes, it involves T cell activation. There's a T cell, huge T cell component. I'm not a believer in this monotheistic view that T cells are better than B cells or B cells are better than T cells. They're all important. You can't get the disease without one or the other. Yes. Yes. The immune system is stupid. Okay? It just sees something's foreign and this co-stimulation make an antibody. Sometimes it's helpful, sometimes it's random. I mean, you don't want to make disease, right? You don't want to make yourself get a pancreas that's going to be hit by antibodies. So you make an immune response because some tolerance might have got broken, there was some cross-reactive antigen, you made a response. Or you did damage the tissue and proteins came out, so you make some antibody. But it doesn't have to be causal. Often it's a consequence of some ongoing pathology, but not necessarily the cause of it. Sometimes it's the cause, but sometimes it's not. Okay, so yes. I will talk about CD8 cells for 5 minutes. It's okay. Yes. Yeah. Abul didn't tell you about dendritic cells?
He did.
Okay.
But we learned something from you also. Actually about, so we know that T cells get apoptosed after the differentiation and the elimination of antigen. Somehow they get and we have legal T cell memory cells. What happens to dendritic cells? Like
They die.
they die. That
Dendritic cells die very soon. They do their thing, and like all good soldiers, they die for us. Okay. Any other questions? Okay. I think I'm just going to go on to the next lecture. We'll save the wrap for the end when Abul is back.